All right, step one of this lab is to collect E. coli cells from a plate. And I've got my plate here. There's the biomass growing on it. And I'm gonna collect them with a sterile loop. And then I'm gonna put them into a solution of sodium dodecyl sulfate. I've got 500 microliters of it in an Eppendorf tube. I'm gonna mix by vortexing to evenly distribute the cells in the solution. And then I'm gonna incubate all of that for 30 minutes in a warm water bath set to between 60 and 65 degrees Celsius. So to get started, I'm gonna need to spark my Bunsen burner. I'm gonna turn the gas on at the valve, and I'm gonna strike to get a flame. Got my E. coli plate in front of me, and I'm gonna flame the loop until it is red hot. And you can see the loop flaming pretty well in the video, okay? I'm gonna cool the loop on the edge of a plate, and then I'm going to use a rocking kind of figure 8 motion. And note that this is a loop, right? Like not a stab. And I'm going to collect a ball full of cells, right? So a little goes a long way in microbiology. You can see here that there's some whitish gray matter on there, and it's about a half a loop full. That is plenty, maybe even a bit too much for this DNA extraction. I'm gonna take my tube of SDS, and I'm going to put the loop full of cells in, and I'm gonna twirl to get all those cells off. Cap. This is contaminated now, so I'm gonna flame it. And then I'm done with that. And I'm gonna turn the Bunsen burner off so it doesn't get too hot in the lab. The cells are really clumpy in here. So there's like a white clump at the bottom, and I want them to be evenly distributed. So I'm gonna vortex by touching to the vortexer. And I check it every few seconds. And now it's nice and evenly distributed, and there are no clumps. This is now gonna go into a little rack, and I'm gonna incubate it in a warm water bath for 30 minutes. And the water bath is over here in the lab. Let me show that to you. Here's our water bath, right? The temperature is set to between 60 and 65 degrees C. I will just take the lid off, and I will make sure my sample is well submerged in water in the water bath. And I'll put the lid back on to ensure that the temperature stays constant. And then I'm gonna time for 30 minutes. So I could do something else in the lab while I'm waiting for this to happen. While the cells are in there, they're in this harsh detergent, and they're heated up quite warm for E. coli, which typically prefers 37 degrees C. And that harsh detergent is gonna damage the cell wall, and it's going to cause those E. coli to lyse or rupture, which is good because we want the DNA that's inside the cells to come out of the cells and be distributed in the SDS solution.